Hi, it's Amanda. I just want to jump on to show you what your plates look like after you do your initial isolation and show you some differences between contaminated plates that still need to go through further rounds of isolation and isolated plates that are, we think, pretty much pure. Okay, so let's start with some stark differences. We see this TD or tube worm, sorry, TW yellow, number 3 here. We have some really nice differences between the white and the orange colonies. You can see the white colonies are a bit larger than the orange ones. I had named this yellow, but obviously it's turned more orange on the plate now. Sometimes the color of the colony can also depend on the type of media that it's grown on. So this is always something really fun to look out for. But what we can see is that there are definitely at least 2 different types of bacterial communities on this plate. And so we would need to then isolate some singular colonies that are maybe, way out there that nothing else is close to, and try to further purify bacterial strain. Now, that is an example of 2 that are mostly the same in terms of like number of colonies representing each. In this case, this is one of the more contaminated plates where you can see that there's quite a bit of orange up, oh my gosh, quite a bit of orange up here, but then this really comes down into white colonies down here, almost like the white colonies are out competing the orange colonies in this case. You would have to select like a single colony as best as you can and then transfer it to a new plate. And sometimes you're not successful in separating them again, as this is actually the case here, where this one was contaminated and then I struck it out again and it was still contaminated. And then I struck that out one more time and now it looks way more pure. So that's exciting. I would feel more comfortable about storing this isolate now. And then we also have some purple colonies that we were looking at. These are the ones that our lab is really interested in. And as you can see, this is maybe the more like white colony dominated example, where there are just little bits of the darker colonies in here. Even though there's some color up here, you have a lot of white coming down here. But as long as you can grab a single colony and then transfer that onto a new plate, you should be able to get rid of the contaminant. And then here's an example where there's mostly purple and then a couple examples of the white colonies like up in this area. And you would still need to restreak that, even though it's mostly the one colony, because once we go on to identifying the different bacteria, if there's more than one type of genomic DNA in the sample, you can get really conflicting results and you'll actually never figure out for certainty what your bacterial strain is, because there's more than one and the computer doesn't know how to tell you that. So the next part that we're going to do is look at the pure strains. So these are the ones that we've been able to isolate for purity, and we're pretty confident that they're totally clean, and we can isolate a single colony of that and grow it to store it or grow it to extract its DNA. And you can see these are the purple ones. If you put your hand behind it, there's no white colonies behind it. You can then take a single colony of this and store it and know that it's just probably one bacteria there. What we can see is the primary streak is really, really dense generally. The secondary streak becomes less dense. And once you get to the tertiary or the quaternary streak, you'll really start to see like full single isolated colonies further away. And as we can see, what's kind of interesting here is that, we almost have this brownish color that is complemented in the purple here. And here we have these like big, meaty colonies, and essentially they're not competing for space at some point. So once you have a bacteria that's really kind of far away by itself, it can grow a little more unchecked, which is really interesting. And something to note of this plate specifically is that, let's see, we have one with a date on it. So right now it's the 23rd, and this was the 20th, so 3 days ago. The colonies are way bigger at this point. The first day that the colonies grow, they come out of the incubator looking like this, where you could almost barely see these baby colonies, but they're still there, right? They're still single. But as a plate grows, even on the bench for more and more days, it gets bigger and bigger, and then eventually it uses up all the nutrients in the media and it can no longer sustain itself. And so you really do have a life cycle, and each bacteria has a different amount of time that it can last on the plate. So this is something really important to know. If you grow something on one of these plates, you need to do something in a couple of days or you're going to start to have your colonies die. And then that means we're going to have to start all over again, right? We have to isolate the bacteria again, and all these things take multiple days. So you really needed to stay on top of in what order you do things and kind of consecutively make sure that you're keeping your bacteria alive, or else you will have to do it all over again. And trust me, it's happened to me quite a bit, you know. So like life comes up and you're like, well, I can't go to the lab this day, but this is grad school, this is lab, this is science, and that's okay. Okay, so the next thing in our next video, we'll talk about what is the next step for keeping these bacteria growing and happy, and how can we store them to make sure that we don't lose them and we don't have to go all the way back to the beginning. And that's on next time.